Hello everyone, welcome back to the Genomics Bootcamp. In this video we will do a tutorial and a showcase of the long-awaited genome-wide association studies. And we will do it on the data of the paper that is described here, the genome-wide association study of deafness in 3 canine breeds. The statistical program that we will be using will be the GEMMA software that is available on GitHub. I use this program for GWAS all the time. I believe is one of the best, but it has one drawback that it works only on Linux. So you need a Linux system to run it. Other than that, it's freely available, and also it has a very nice description. So if you just put in GEMMA GWAS in Google, then comes up with this GitHub page. Actually, when you scroll down, there is a very nice description of the program, so you have all here, including a very nice manual, and most importantly the pre-compiled binaries. So you just download these and you are good to go. This is of course a requirement for this tutorial as well. Before we do the actual tutorial, I just want to tell a few words about the theory so that you recognize what's happening in the code. I talked about this in my theory video, the Introduction to Genomics Part 6. So I'm not going to tell too much here. So if you want to get more details, I really suggest you to look up this video. So very briefly, in genome-wide association studies, we search for the regions of the genome that influence the phenotype of interest. You can do this based on SNPs or sequences. In this case, we will be doing it via SNPs, which is the most common procedure. So what you want to do is estimate the SNP effects and their significance. Here we have many possibilities. In this case, we will do a simple linear model. We want to get something like this, but how to get to this picture, so this will be the topic of this video. There are two very important things to do, and the first one is to account for the population structure. In the theory video, it was demonstrated with two populations of mice. So basically, we have to account for the structure of the population before actually running the computation. And the second thing is that we want to avoid false discovery. So basically, we do not want to have all our SNPs sky high, because then we do not know what to concentrate on. So basically we want to get rid of the false positive results, and one of the most used ways is the so-called Bonferroni correction, where we divide the usual significance level, that is usually 0.05, with the number of tests. So this we will be doing within our script. So then you will recognize it when we will be doing the Bonferroni correction. As I told you, there is a much more detailed information in the theory GWAS video. So if you want more, perhaps look at that video before continuing here. Also, as I mentioned before, the two most important things that you need to have for any GWAS study are the genotypes and phenotypes. In this video, we will use the open access genotypes and phenotypes from this canine study. But for any new runs, you probably want to have your own data. And when it comes to generation of new data, we very often go with the services of the NEOGEN Genomics, who are also very kindly supporting this video. So I would like to take about a minute of your time to introduce the company, and also then introduce a very easy way how you can get a £100 reduction on your order. So NEOGEN as the company provides an extensive range of cost-effective customized genomic solutions and services for any agricultural species. With leading commercial genomic laboratories located around the world, they deliver a consistent service with rapid turnaround times, empowering your decision-making in genomic selection, health management, and research. NEOGEN's high-quality array portfolio provides efficient testing to develop targeted predictions and research opportunities at an affordable price. Their custom GGP chips combine outstanding design and even coverage of the genome with SNP parentage marker content and causative content. Key drivers behind all of NEOGEN's new chip designs include unique SNP select software and a multiple objective local optimization or MOLO algorithm, enabling them to choose the best set of SNPs with optimal genome coverage, recombination rates, and minor allele frequency representation. What's more, NEOGEN routinely genotypes on Illumina and Thermo Fisher public consortia and custom arrays on behalf of their customers. The future of genomic innovation is here with NEOGEN's DNA sequencing capabilities. NEOGEN has been a leader in the innovation of whole genome sequencing, which offers the most detailed view into your genetic program by evaluating every base in the genome and navigating the complexity of genomic variants that make individuals unique. NEOGEN also provides skim sequencing with imputation for select species to augment genomic selection strategies currently in place, in addition to a new technology called InfiniSeq, combining NEOGEN's world-class laboratory services with GENCO's low-pass sequencing software and advanced analytics platform. InfiniSeq delivers the necessary insight to confidently select superior individuals from breeding populations and research objectives. And here we are at the mentioned offer. So as for you, for the viewers of Genomics Bootcamp, NEOGEN would like to offer a £100 off a £1,000 spent on genotyping of their GGP arrays. To access this offer, simply email lperry@neogen.com with the subject line Genomics Bootcamp. So you can access this offer and get all the details. As you see also on the screen, there are terms and conditions that apply, and well, you can read those on the screen as well, but also I will copy everything to the description of this video, so you can also just copy paste, especially the email, and check out the possibilities here. So once again, I would like to thank NEOGEN for supporting this video and their overall friendship towards the Genomics Bootcamp channel. So back to our tutorial with the data on deafness in dogs. At this point, what you should have is the data downloaded from this paper on a Linux machine, the GEMMA executable file with the compiled version or compiled form, and also Blink, because we will use Blink for the data preparation. The overall working environment for the entire process will be R as usual, so well, you should have R installed on the machine that you are working on. The whole script is, well, the start of the script is on the screen and it has several parts. I will walk you through, and yeah, basically we will show what happens in each of these parts. Funnily enough, the GWAS itself is just a very tiny part of this entire script. It's basically a single line. But as always, there is a lot of things that you need to prepare or do before actually doing any kind of analysis. And if you want to visualize the results, also that takes a bit of a scripting. So that is also included already in this R script. So the parts of this script, as I mentioned, so there is this introductory part with, well, just cleaning the workspace. There are some packages that you will need. So if you don't have any of these, then, well, you need to install them within R. Then there is a very specific part for this actual run, and this is the preparation of the phenotype data, because it is in a, well, a specific form, and we want to get it into a form that is usable by Blink. So this part is just for this data. The next part of the script is the preparation of the genotype data. This is already in Blink, So this involves quality control and also updating the phenotype column. So I will get you through and walk you through all this more in detail. So this is just to show the parts of the script. The next part is running GWAS with GEMMA, and this is what I mentioned. So basically the running of the GWAS itself is just one single line, and then before we do the kinship matrix for the mentioned population structure correction. And then at the very end we want to visualize the results, and we will do this with R, and also an optional part if you want to extract the results that you care about. So if you don't want to look at, I don't know, 50,000 lines, then you can easily extract just the highest or most significant SNPs. So the first thing to do is to prepare the phenotype data. So as I mentioned, this part is very specific for this data set, but it just shows what you need to do in order to do this GWAS run with GEMMA. GEMMA has a huge advantage that it actually uses Blink files directly. And you might remember that the last column of the FAM files is the so-called phenotype. And most of the time when you have the FAM file as shown on the screen, so in this case there is deafness FAM, so that you have the family ID and the individual ID, and there is the sire and the dam and the sex information, and the last column or the sixth column is the phenotype. And most of the time it's well, -9 or 0 or basically unknown. If you are able to get the actual phenotype information to this column, then you are all set, because then GEMMA could just use this information for each particular individual for the run. So this is what we want to do. Now there is a bit of a problem, because the phenotype information comes like this. So it's a file, very nicely done. But so there is this dog ID and the sex, but there is also the well, the breed information, and the phenotype itself is either hearing, bilaterally deaf, or unilaterally deaf. And for Blink, what we need are numbers. So basically we need, categories 1, 2, 3, and actually also missing if there is any. So we need to transform this file to a file that looks something like this. So that has a column FID and IID for the family ID and individual ID, and then one column for each phenotype, or just one column of phenotypes if there is just one phenotype. But anyway, so it has a name. In my case, I named it deafness code and deafness case control for case control study. And well, this is how I called them. So I renamed and recoded the categories of the hearing, unilaterally deaf, and bilaterally deaf dogs. So, and how to create this file, which is usable by Blink. So this is the first part of the code. So this first part has this phenotype that we will use later on for our GWAS run, but well, basically not so important right now. The line here with the read underscore table is reading in the deafness underscore pheno.txt. That is the phenotype file provided by the authors of the paper. And then with the combination of the tidy versus mutate and case when statements, we recode the bilaterally deaf as 1, unilaterally deaf as 2, and hearing dogs as 3. So this is for the column deafness, which I call deafness code. But then we could use also the opportunity to create more phenotypes columns. So for example, for the deafness case control, I decided to recode the bilaterally deaf dogs as 1 and hearing dogs as 2. And therefore the unilaterally deaf dogs are appearing as missing. So we have then the FID and IID columns also created, and writing out the GWAS underscore dog underscore pheno.txt, which is basically this file. If you have any other kind of data basically the first step is that you get the file similar to this, which has the first column FID, second column IID, and then at least one column with your phenotypes, because this file will be used in the next step to update the phenotype column with Plink. And here we will do just that. So the first thing that we will do, as usual, is the quality control. Now, in the paper, they use a slightly different quality control measures, but basically it was about the usual stuff. So basically deleting the individuals and SNPs that have too much of a missingness. And then we are create a file that is after quality control, and then we update the phenotype. And what we could do this with using the --pheno Plink option, where we include the file GWAS_dog_pheno.txt. So basically this file, so the file that contains all our phenotypes for each individual. And then we combine this with the --pheno name, and that is the name of the column that we want to use for the GWAS run. Here I have it written just phenotype in all caps, and that's because I used it earlier as a phenotype and assigned it as a deafness case control. So as you see here, so this is one of the columns that are in this phenotype file. The advantage of this method is that, with a little bit of tweaking, you can do loops basically with GWAS runs. So you can run many GWAS runs for each column of your phenotype file. But well, let's leave that to another day. So this time we will do a single GWAS run. So to summarize this part, with the --pheno and the --pheno name, we update the Plink file. So now we have our phenotype column filled. And then for the sake of the tutorial or just demonstration, we just select only the Australian cattle dogs, so we can directly compare our results with the paper. So from the pheno data, we find out the FIDs and the IIDs of these Australian cattle dogs, and with the --keep statement, we just create a file that will be an input for our GWAS. So again, this is purely for this example, so that we have a direct comparison to our output graph with the paper. And in the next step, we are actually already at the GWAS run, and considering that how much time we spent already on all the explanations and preparations, this is almost like an anticlimactic outcome, because it's just so simple. So basically the whole GWAS with GEMMA in this very simple and very basic form is the following. The first line, we are using the GEMMA executable file. So this is actually in our working directory. And as I mentioned, we are at the Linux system. So basically we start with the // and an executable file name. Then we have -b file. So just take care, not double dash as in Plink, but in just -b file, which is like a GEMMA option. And we put in the input file name for our Plink file. So here we created this input for GEMMA _ACD for the Australian cattle dogs. And this will be the input for GEMMA. This -gk is basically does a relationship matrix and computing the kinship, and the output of this will be saved in the file that will be with the relmat name. So basically in the output directory that will be automatically created, there will be a relmat.cxx.txt file that we will be using later on. And the later on is right after, actually, because the actual GWAS is run in this line. So we execute the GEMMA again with the /GEMMA and the exact file name. This file name actually might change depending on which version of the GEMMA you have, but this is the recent one that I've used. Again, we are starting the very same way, so we say -b file, and the input file name will be also our plink file that has the phenotype column filled in. We consider the relationship matrix with the -k option. So this is to consider the relationship matrix as a measure of population structure correction. So I emphasize many times that the population structure correction is very important and very necessary for genome-wide association studies, and actually this part, this -k and the actual file with the relationship matrix takes care of this. As I mentioned, for this case we will do a linear model, so there's the option, so there is a number of options what you can do with GEMMA. So we will do a simple one right now with the -LMM2. So this is one of the options for this linear model, and also then the -O for the output file name, basically the GWAS result.lmm, which is, this is what we will be using, or well, this will be the name of our output file. So, and if you run this, it's well, with the linear models, it's quite quick. So you will have your output file in no time, also in the output directory. So, and basically that's it. That's the whole GWAS for the execution part. So, and this is how the result file looks like. So, well, there is a bit of a shift in the columns, but basically you have one line for each SNP, and well, there is the chromosome, the RS, so the name of the SNP, the position, the number of missing, allele 1, allele 2, and also some other statistics. But the one that you want to look at least in this particular setup, is this last column that is basically the p-value for these, or for each SNP. So for each line there is a one SNP, and at the last column is the p-value that actually shows the significance of the SNP in that particular run. And of course, well, you don't want to look at, well, in this case, almost 140,000 lines, but you want to look at a nice Manhattan plot, so you can see straight away what's happening. And of course, you want to do that by visualizing the results. So this is the next step. Fortunately, we have well, R, and that can take care of things in a very quick way. The first thing that you need to do is to read the GWAS results. So we do this with the read_table, and the actual file name for the GWAS results. So as I mentioned, that is in this case, because the output file name I put in, that was GWAS_result.lmm.assoc.txt. So this is the files, or the file with the results. I also mentioned in my introduction that you want to get rid of the, or actually take care of the false positive results. So you can do this with the Bonferroni threshold. And this is also, well, fairly easy to do. Because what you want, your Bonferroni threshold is the -log10 of the 0.05. So this is the common significance threshold, and you divide it by the number of tests. And the number of tests is basically the number of lines in your result file, because as you remember, so there is one line per SNP, and basically for each SNP there was a test or a linear model run. So basically your number of tests in this case is the number of rows in your output file. Here I would like to also underline that, well, there are different ways how to compute this number of tests. So some people, and I believe also in the paper, it is done in a way that they are considering like LD blocks or haplotypes or all this kind of stuff. It's also possible. But for the sake of simplicity here, I assume that the number of tests or number of independent tests that is being run is the number of SNPs. Again, as for many of these things here that we are talking about, there are many options. So I'm no way even suggesting that this is the only solution that can be done. But anyway, so what we do is divide 0.05 as a significance threshold with the number of tests, take a minus log 10 of that, so we can put a threshold on the Manhattan plot. And the Manhattan plot itself is done with the very convenient Manhattan function. And this Manhattan function comes from the QQman package, which is really great for this purpose. So basically what you need to do is, well, the Manhattan as the name of the function. So the results GEMMA is basically the file name, or actually the dataset name that we used here when reading in the GWAS results. So this is the results, GEMMA is the name. So the chromosome and the base pair position and the p-value. So you need to get or give the exact column names and also the SNP name, and we want a genome-wide line. So basically we want a line with the Bonferroni there, which we calculated earlier. And we envelope the whole thing in a PNG and the dev of R functions to create a picture directly on the hard disk. Here I would just mention that although I really like the GGSave as a part of the tidyverse, for some reason in this particular script it didn't work for me. I don't know if this is issue with my server or the server that I am working on or something else, but it was not working. So I opted for the base R, PNG and DevOf solution, which worked very well. And of course, with the QQman package, there is also a possibility to do a QQ plot, where the very same way with the function QQ, and here the only input that is required is the column of the p-values. And again, this is put directly as an output, as an image. And as I mentioned, if you want to look at just a few lines, or well, relatively few lines that are above a certain threshold, you can do it very easily in R already. So here you use or make use again the result GEMMA dataset that you loaded previously into R. One minor thing, you need to calculate again the neglog p for all the lines. So just to to have it there, because it's originally not there, because it's in the visualization is calculated directly in the Manhattan function. So you just create a new column and then you filter according to this negative log p as you wish. So I put it that the outcome should be the ones that higher than 5. But of course, this is very much on you, what threshold what you want to implement and which SNPs you want to see in detail. And after you run it, so you get this GWAS plot. So this is a direct comparison with the paper. So you see the same peak on chromosome 3. Well, there is some differences between the two graphs. The first one is that mine is much less pretty because, well, this is just a quick graph. So definitely it could use some visual tuning. And also, you might notice that the threshold is in a different place, and that's because, as I mentioned in the paper, they used a different number of tests that led to the setting of the significance threshold. So whereas I used all the SNPs, in the paper I believe they used something akin to LD blocks or something like that, so that's why the significance threshold is slightly different, but in general you see the same outcome. And this is basically the place I would like to wrap up with this basic GWAS tutorial. If you have any questions, be sure to ask them in the comments below. If you're interested in the exact script, I will put that online on my GitHub page, and you will have a link in the description of this video, as well as to link to the paper and the data, so you can actually use this dog data also for practicing. If you're interested in GWAS in a greater detail, be sure to let me know, and I will try to expand this topic with a few more videos. For now, however, I thank you for your time and wish you a really nice day.